negative, which means what? It tends to soak up electrons, right? It tends to attract electrons. And that's exactly what's happening here. You can see the electrons are being attracted more over to the oxygen side than the carbon side. So the oxygens are becoming more reduced and the carbon is becoming oxidized. So that's what happens. So same thing occurs kind of during respiration. You can think of this as like glucose being burned or combusted with oxygen. And so there's an oxidation reduction that's going on here as well. But in this case, what is being reduced and what is being oxidized? Well, go back. Right? Oxygen is being reduced here because it's soaking up electrons. So it is called an oxidizing reagent or an oxidizing agent. Okay? It's basically causing oxidation of the carbon here. And so, what's going to happen here?
The oxygen gets reduced and the carbon gets oxidated.
So same kind of deal. And so oxygen, again, is acting as an oxidizing agent. And then the glucose is going to be reduced. So there's a lot of oxidation reduction steps that are occurring in respiration. That's why that introduction. And so one of the things that we would be interested in if they're oxidation reduction reactions is where do all of the electrons go? What happens to them? So there are three main parts to respiration, or at least your book calls all of these things respiration. Some people just refer to this as respiration. That's just terminology. So the first step is glycolysis, which means splitting of a sugar. See how glucose gets split to make pyruvate. And in the process of that, ATP is generated and electrons are released. So the high energy electron states are captured by an oxidizing agent that's called NAD+, and it's carried as NADH. So those things are going to come down into here and help make more ATP. And there's this thing that's called the citric acid cycle. This is what Albert was saying Georgi was contributing to, knowledge of. It also makes ATPs. This is where carbon dioxide is actually released. And again, more electrons are generated and carried over to this process, which makes a lot more ATPs. So, first let's just look at what these electron carriers, what that look like. So as I said, electrons are carried by this molecule NADH. What is that? It's the reduced form of NAD+. So NAD stands for nicotinamide adenine dinucleotide. So what is that molecule? What does that look like to you? What's this?
So the carbon ring structure.
Yeah, okay, it's a carbon ring structure. And what kind of carbon ring structure? Adenine. This is the base. Right? In DNA, or RNA, actually, in this case, because this is what? You recognize this?
Oh, ribose.
Ribose, that's exactly right. So it's got 5 carbons, and they're arranged in a spring-like structure, and there's an OH here. So it's not a deoxyribose, it's just ribose. And then this is attached at the 5' carbon here to a couple of phosphates. But this phosphate is attached to this 5' carbon. So this is like a 5' to 5' This is not what you will find in RNA, right? That's, in RNA, it's usually 5' phosphate 3' to the next OH. And then over here, this is attached to another weird base. That's called nicotinamide. So this is actually derived from the vitamin B complex. So as I said, this is the oxidized form of this molecule. And so if it's reduced, then electrons are going to be added, okay, to this. And because electrons are added, you reduce the charge from a plus to no charge. How does this occur? So actually, these electrons are being carried by hydrogen atoms. Okay? So the hydrogens donate their electrons to this form, and also a hydrogen. So oftentimes, when you see these oxidation reduction steps, you'll see the addition of a hydrogen to one of the molecules. And that's called a reduction because, not because necessarily because of the hydrogen itself, but because of the electrons that it's carrying. And then a release of a proton into the solution. So this is called NADH because of this addition of that hydrogen. And of course, this is a reversible reaction. So when you oxidize NADH, you get NADH. Any questions? Yeah.
So when the two H's are added, there's two electrons, but then only one of them hydrogen. So
Exactly right. Okay. So those are those molecules. Let's go down to glycolysis and look at what's happening here. So there's a lot of details about this in your book. Those details, of course, very important, but for this course, this is an introductory course, and we don't expect you to memorize all of those details, like where each hydrogen comes from, and what the structure of succinate is, and malate, and all those different kinds of things, what the names of the enzymes are. What we do expect you to understand is what are the main inputs into this process, what are the main outputs. Why is the pathway constructed this way? What are the main regulatory points in this pathway? And how does this pathway actually provide energy to the cell? Right, so those are the main kinds of points that we would address on an exam, for example. So this is the first part of the glycolytic pathway. And in this first part, there's energy that's actually invested, and it's invested through ATP. So there's two molecules of ATP that are actually expended to get this process going. Right? This is like, Wall Street. You need to have money in order to make money. So this is the money that you put in first, and then you're going to get a return on your investment later.
Also, other people's money instead.
Other people's money. Okay, well, you can also invest your own money. So glucose is a 6-carbon sugar, obviously, and this ring, and so it is a hexose, and so the enzyme that adds a phosphate group to this glucose molecule is called hexokinase. Kinase is add or sometimes take away phosphate groups, right? And they change the phosphate group around. Often that involves ATP. So now you have a molecule that's called glucose 6-phosphate because it has a phosphate on the number 6 carbon up there. And this is catalyzed by this enzyme hexokinase here. So there's a little pocket in here, which is the active site for that reaction to occur. Here's the next step.